this summer. Thank you.
Thank you. Thank you for the introduction. Again, my name is Bill Webb, and I worked with Sam this summer as part of the group that is in the Shell Lab and the Shea Lab, and appreciate several of you coming this morning. I've seen you in some of the meetings that I was able to attend. Appreciate you inviting me to those meetings and hearing about the great research that you're doing.
And we've got some Zoom folks joining us as well.
Oh, okay, great.
Thank you.
Yeah. I currently teach at Oaks Christian School, which is in Westlake Village, about an hour and a half from here, and I've been there for several years teaching physics and engineering courses. So this summer was definitely, as Frank mentioned, a stretch for me because I'd done very little coding, and that wasn't necessarily the only thing that I did this summer, but it was a major part of it, especially early on, developing codes and writing codes. And so that was something that I had to learn a lot about. Also, a lot of the different visualization software programs and other software programs that I had never used before, there was a learning curve. So the first 2 weeks, it was primarily just Sam tutoring me and helping me learn how to use these tools. And then the next 2 weeks, it was, okay, now getting the code perfected and gathering some data. And then these last 2 weeks, okay, what is this data telling me, and trying to make some sense of that. So I appreciate Sam's patience through the whole process. And what I was analyzing and learning about is how basically to do computational analysis, which I'd never done before, and focusing on the COVID virus spike protein and how that connects to the ACE2 receptor in humans, and specifically how the hydrophobicity of that spike protein might change from variant to variant of the different types of COVID, and how that might affect infectiousness or disease symptoms. So that's in a nutshell. You said there was something that I needed to do to, or just, okay, here we go. All right. So just quickly to overview the big picture, the spike protein on the SARS-CoV-2 virus does connect to the ACE2 receptor in the human cells, and that's one of its methods of infection. And that's the one that we were focusing on for this part of the research. And then this is just a PyMol visualization to help you kind of see how they connect. And I've intentionally kept them in two different views. So the one on the left is the binding of the spike protein for the SARS virus, and the one on the right is the ACE2 receptor. And I've left them in different formats, one the cartoon on the right and one the spheres of the atoms on the left. So you can see the alpha helix that kind of comes through this groove in the spike protein, because that's one of the things we were focusing in on. And on some of the visualizations that I show you later, we'll be looking basically from the perspective of this ACE2, looking at where it's going to dock basically on that spike protein. All right. For proteins to bind, one of the steps that has to happen is the water that naturally covers the whole protein, including the binding site, that water has to be pushed out of the way so that the binding sites can come together and actually connect. And so that de-wetting process, that removal of the water, is kind of a big focus for what we were trying to analyze. Is if, for instance, the binding site of a particular COVID variant is more hydrophobic, meaning it's going to repel waters, then it would more easily push those waters out of the way and let it bind more easily. However, if it was hydrophilic in nature and attracting waters, it would take more energy to push those waters out of the way and be less likely to bind, or bind less tightly. And so that was a major focus, is understanding that hydrophobicity and zeroing in on where they bind together and analyzing the hydrophobicity in those areas. Now, the big objectives for the project for me, in terms of what I was hoping to learn once I started analyzing the data, is could this hydrophobicity help us begin to understand why was Delta more deadly? Why were the symptoms more serious? Why was Omicron more infectious? It was easier to transmit. What allowed different variants to have differing immune escape values? And then also even start to think about, could we predict what the next mutations might be? And then even thinking about what vaccines might be necessary to counter those new mutations. So those are some of the things I was hoping to sort of investigate along the way. One of the articles I read as I was researching by Patel from a few years ago, he said that a characteristic of protein hydrophobicity, which informs its interactions, has remained elusive. And there are ways to measure hydrophobicity, but most of them focus on studying the protein itself. And most of them don't have a lot of context. They just say, well, serine is always going to have this hydrophobicity and valine is always going to have this hydrophobicity. And they don't really pay attention to what's beside each other in the sequence, and then how the secondary folding occurs and how that might affect it. And so our goal was to improve on those methods to make it more contextualized. So we studied the water versus the protein surface. Now, water in pure form naturally hydrogen bonds molecule to molecule and forms this tetrahedral shape, where the angle between every three waters is 109.5 roughly degrees. And so regardless of which three waters you pick, you're going to get that same angle if it's pure water and there's nothing changing those angles. And so what we were doing is analyzing triplets of water molecules and finding out for the two layers of water that surround the protein, what were the actual angles? Because they wouldn't probably be always 109.5 anymore, because now there's going to be hydrophobicity and hydrophilicity to consider. And so those angles change in those conditions. So for instance, if this is a close-up view, we were using molecular dynamics analysis to get computational data. We have this valine amino acid and two layers of water molecules all around it. We would look at, for instance, those three waters and figure out what the angle is. And the computer is, through the analysis, determining all these angles. And then about these three waters, and then those three waters. And in this visualization, it's just showing the oxygens, because if the hydrogens are there, it just gums up the visual. So, but then we've got, again, those three waters and so on and so forth, just analyzing the water triplets, what's the angle between them, and how is it different from that 109.5. Now, once we would get that abundance of data from all the amino acids, we could graph it. And this is just one portion of amino acid residues and how they would graph out in a line graph that's representing really histogram data. What is the abundance or the probability distribution of angles that are in this 45 to 50 degree range, which tends to be the more hydrophilic nature when the angles between the waters are in that range? That's going to be a hydrophilic signature. Or versus when they're closer to that 109.5, and that's going to give us the hydrophobic signature when we see a lot of angles in that realm. And one of the reasons we want to use this graph is this shape is pretty typical, and we could kind of compare our data to make sure if it seemed to follow the right data, make sure it's sort of a sanity check to make sure that we were, our data was meaningful. Now, the reason that we were using computational analysis, obviously you can't just look under a microscope and see all those angles. That's a big reason, but there's other valuable things about computational analysis compared to wet lab, although they're both incredibly important. And one thing that really impressed me is how well the groups in the Shell lab and the Shea lab coordinate, meeting every Monday, sharing their thoughts. Some are in the wet lab side of things, some are in the computational side of things. But in the computational, some of the advantages in thinking about how it might be shared at the high school level with my students is you're going to have less disposable and consumable costs. There's still costs, but not in that area. Then you're also going to be able to get data in a very efficient time frame for a lot of samples. It also, you don't have the same legal and safety regulations you would have in a lab. And for my students specifically, if they learn a little bit about computational analysis, that's a marketable skill they can put in their applications for college. A lot of the research that revolves around studying COVID focuses in on the electrostatic charge distribution. And just that's the element that they're trying to investigate to see why are the different variants different in their effects. And there are some that focus in on hydrophobicity, but as I mentioned before, their models are pretty context-free. They just say, well, this is an alanine, so it's going to be that. Okay? And so if you look at this example, I've put little yellow circles around the two places where the amino acids are mutated in the spike protein bonding area. And if you look within the circles, you can see there is some color differences there. But when you look outside of those circles, it's virtually identical. And that's not what we expected to see when we get more context to the picture with our analysis. Now, my workflow was I would get the trajectory files that Sam would produce for the various forms of the COVID virus and visualize a movie so I could sort of see if it made sense, that was another kind of skeptical check, and then take a Python script that I developed that sent batch files to the cluster, computer cluster on pod, and then get all the data for me. So this is just a quick animation to show you how looks like it's having trouble on the Zoom, but you can see the water molecules are moving around the spike protein, and the MD analysis is taking those water measurements all in 2 layers deep all the way around the protein. It's a little bit hard to see on that screen. It's a little smoother on this one, but hopefully you can get a little bit of an idea about how that animation works. And then, like I said, I would send those batch files to the pod cluster, and it would bring me back a bunch of data. Then I developed another Python script that would take that data, store some of it in a data frame for manipulation, store some of it in CSV files so I could access them later and do further analysis. And I would have scripts like this and data like this. And so just to give you an idea of the things that we're looking at behind the scenes. As I said, it's important in computational, in any lab work, but especially in computational work, to make sure that you're being skeptical. You don't want to just say, oh, this is telling me exactly what I thought, without actually digging in and making sure your data makes sense. And so along the way, we found some things about the bulk water aspect of it. There were some coding issues where we had to change a few things or just improve some things as well. And then I take those CSV files and those data frames of data and turn that into graphs and other visualizations. That was kind of the final step for analysis. And these are just some of the different kinds of graphs and visualizations. And if you're curious later, I don't really have time to go into all the details in this short presentation, but I can talk to you about some of them. The main ones that I focused on primarily were these two on the bottom, where I'm looking at the hydrophobic signature, what abundance of those hydrophobic angles are showing up, and then the hydrophilic, what abundance of the hydrophilic angles are showing up. And then over there, we turned that data into de-wetting free energy and could subtract SARS minus the other variant to see which one was more hydrophobic and hydrophilic by subtracting those values. But the one that helps us kind of visualize it is taking that information of the hydrophobicity, hydrophilicity, and the de-wetting free energy, and then putting it into PyMol to visualize atom by atom and amino acid by amino acid. Where does it show a more hydrophobic part of the binding of the spike protein? Thinking about the different mutations, there's the ancestral SARS-CoV-2, and then alpha just had one mutation, then beta had a couple more, gamma just had one slight change from beta, and then delta seems to have derived some other way because it has different mutations than Omicron, very different mutations in many of them. So just to kind of give you a picture of what those differences are. Today, I'm just going to focus quickly on two, Delta and Omicron. And so if you haven't looked at these visualizations, I'm mostly just showing you 2D visualizations, but you can manipulate them. And so you can see them in 3D, you can rotate them in any direction, you can dig in and dig out and zoom in and zoom out. And so what we're seeing here is the red color indicates hydrophobicity, the blue color is hydrophilicity. And then one of the things we've improved the code with is we started to see that some of the amino acids had very few water triplet angles actually in the data. And so if there are only a few or no angles measured, they must be shielded from the water in the analysis. And so we filter those out, and that's what the blacks are. And you can tell from the animation that those are mostly buried. And so they had fewer than 10 angle measurements, where most of them were 70, 80, 100, different angle measurements. So if they were so few, it seems statistically irrelevant and maybe misleading. So we filtered those out. Here's another look, again, just focusing on the two mutations from Delta on the right to the ancestral on the left. And you can see, again, for Delta and the ancestral, in the circles, there's a difference. But what we thought might be true is that Delta would maybe be more hydrophobic because it was more deadly. And what we see actually is that it went from pink, kind of more hydrophobic, to more white, less hydrophobic in the 478 transition. And then in 452, it went from pink to white again. So actually for Delta, it didn't follow what we kind of expected to see. And when you look at the rest of it, there's, again, not too much difference. So the context-free analysis didn't really show much different than what we found in Delta, at least. However, that's not necessarily been the case for the others. One of the others is Omicron, and there were a lot of mutations. So I've highlighted in yellow all the different mutation sites. So you can see how many of them there are. Again, we're looking at the COVID spike protein from the perspective of ACE2, like where it's going to dock. And so you can see a lot of the mutations are there on that part of the spike protein where it's going to connect. And if we look at the hydrophobic and the de-wetting free energy and what that tells us about the hydrophobic nature and the hydrophilic nature, if you kind of focus first in on the box right there, you can see that there are some changes where this white becomes a little more pink, a little more hydrophobic, and this blue becomes white, which is more hydrophobic. And there's some blue here that becomes pink over there that's more hydrophobic. And so in that area, we are seeing in Omicron a stronger hydrophobicity. And then when we look down in this area, we see some similar comparisons where this kind of gray becomes red and And so there's some changes that in Omicron, just visually, we can see it does seem to be a little bit more hydrophobic. And if we look at the graphs, again, I'm not taking the time to dig into all the graph data today, but I have some if you want to see it, but we could see that mathematically visualized as well, beyond just the molecular viewing. So for me, the conclusions I was able to draw in this short fire hose of a summer, one is that the water triplet analysis, that form of computational analysis, seems to be a valuable tool to try to quantify the hydrophobicity in COVID variants. Next, the hydrophobicity model is computationally cheap compared to some of the other models. Sam was talking to me about some of the other research he and some of you have been involved in that use that computation that costs a lot more time to do, whereas many of these analyses would run in just a matter of minutes. Hydrophobicity doesn't seem to be able to explain the difference between the original SARS-2 with Delta, based on what I showed you, but it might be an underexamined factor when trying to understand Omicron's infectiousness. So it might be something that needs to be more further investigated. So those are some of the conclusions I was able to come to. And again, we've just in the last couple of days, Sam's really been digging into alpha and beta and gamma and seeing some really interesting things there too, but not enough for me to digest and sort of try to explain here this morning. Some future research opportunities I think would be important to consider. That de-wetting free energy equation, or that model that was used to take the hydrophobic and hydrophilic data and then convert it to a de-wetting free energy, that was taken from other research that Sam has done. And so it might be worth taking some time to see if that needs to be tweaked at all for COVID specifically. And then one of the things that Sam has been doing yesterday and the day before, just kind of a more granular, looking deeper, okay, not just amino acid by amino acid, but looking at them in smaller clusters instead of big picture. I think that further analysis might be interesting and fruitful. A statistical analysis of the data to determine significance, and he just sent me some things this morning about that. He continues to dig into that as well. Just something we ran out of time for my particular involvement. And then validating and correlating our model of using that triplets with the binding constants for the different viruses to see if that lines up. And then the big question that I think is really interesting, could this model and measuring hydrophobicity help predict what future mutations might be and vaccine development? In terms of curricular ideas, one thing I'll be doing right away in the fall is challenging my students who already have a coding background, because some of them have taken multiple coding classes at my school before they're in my class. And so challenging them, hey, can you make your graphs and figures using Python instead of Excel or Desmos or something like that? I think that'll be good preparation for their futures potentially, and a challenge for them. And then I was talking with Dr. Balakrishna here at UCSB about a possibility of cooperating with her to do a computational analysis introduction. There's a special Friday we have in our school year in March where we get to just teach a 3-hour chunk of something we're interested in. And she was talking to me about some modules that I might be able to use with my class for that, to just introduce the concept to high school students. And then I don't particularly teach biology, but I might be co-teaching a biotech class in 2024. And so either sharing these last two ideas with biology teachers at my school, including my wife, and then possibly in that biotech class, using PyMol, having students understand how they can view these molecules in a different way that maybe they never have before. And then the AlphaFold AI predictive software to say, hey, what if we mutated this one amino acid? How might that change the secondary structure of this protein? And letting them play around and experiment with that. So those are some curricular ideas I have at this point. I'd like to specifically thank Sam for all the time you invested in me this summer. The way you scaffolded my learning was incredible, and the time you invested was really appreciated. And thanks to Dr. Shell and Dr. Shea for letting me be a part of it and stealing Sam away from some of the other things he's doing, and just letting me come to your meetings and getting to meet several of the others of you in those meetings and in the lab. It's been a great time. Thank you, Frank and Marilyn, for all your feedback and input, and also Nicole and Laura for your input as well. Appreciate it. Questions? Yes.
Yeah, really nice talk. Really exciting stuff as well in terms of I don't do extensive amounts of biology work, so I was curious. You talked about the angles 40 to 60 degrees is hydrophobic versus or hydrophilic. Yeah, and then 120 is hydrophobic. Could you explain a little more why that is? I'd never heard that.
Yeah, I don't fully. biology is something that I've studied, but it's also not what I spent most of my career working on. And I don't necessarily understand fully why those angles are, but just knowing that from the research that we've looked at, that seems to be the range of angles where it shows that hydrophilic nature. I'm not sure what it is about the chemistry of those angle changes. Yeah,
Yeah, so I understand.
Yeah.
Thanks.
You've met three-body angle stuff. That goes beyond just the domain of biology. But
Sure.
you can, whenever the water people are talking about this, everyone is confused. They're not, Sally and Dennis are just can't talk about
I mean, the truth is we don't super know, but it's empirical. Like it correlates well with some of the other measurements, and it like correlates really well with small hydrophobes. So we took it.
Yeah, that's okay.
Yeah, it's more about like applying the model than validating, but yeah, the validating stuff is pretty.
I'm just sort of generally curious if you were able to attend Python workshop that we had early in, and if that was any help for some of the things that you did.
Yeah, I had never used Python before May. And in May, I just asked my teacher at my school, hey, can I jump into the AP Python class and just like study for a few weeks? And so I did that. And then I came and did the boot camp here for 2 days. And it was great because I learned some of the basic ideas on my own at my school, but then they were focusing on how do you create these graphs and figures. And that was what I needed to understand more than what I learned before. So it was terrifically helpful.
I'm sure Linus would also be willing to share things with you if you wanted to actually talk to your
Okay.
students.
Yeah, that's a great thought. I saved everything he sent us.
Yeah, yeah.
So he sent us some really cool Jupyter notebooks that kind of walk you through steps.
Just to review the really clearly you saw way more hydrophobicity in Omicron. And I think you said this, but that I remember that being famous for being way more contagious, correct? That's pretty remarkably more. Yeah, okay. You had the objective slide as well, and there was some pretty big ideas on there, like
Yeah.
possibly
Yeah.
future, trying to predict the future almost
Right.
in terms. Do you have any comments on any of that after just 6 weeks looking at this? Is that
Well, because it's computationally cheap, you could just take, a weekend and run a bunch of data, like all 20 amino acids. What if they mutated? Which ones create the greatest hydrophobicity? And you could just go through the important parts of the spike protein and analyze it computationally and find out, well, which ones lead to greater hydrophobicity? And then those would potentially be the ones that would be most likely successful to evolve. That would be kind of the thought behind it. Whereas just waiting for it to happen, this gives us an idea to maybe start to get a little bit ahead of it predictably. Obviously, there's no guarantee in the predictions like that, but it'd be interesting to see if it could start to, highlight some areas that maybe we should be concerned about. Yeah.
Is there more evidence in other infectious diseases that hydrophobicity does make these viruses more infectious?
That's a great question. That's not something I looked into yet, but I don't know. Have you looked into that at all, Sam? Or no? Okay. Yeah, that's a great question.
probably it's like, I guess it's like electrostatics, hydrogen bonds, that can be biology uses. So yeah, surely it has that.
Yeah, I just think it's fascinating that what you just said about Omicron.
Yeah.
How it's more infectious. Striking the hydrophobic. It would be cool to see more examples.
Yeah. Yeah, it's something that I'd never thought of before the summer at all, but it does get you thinking about the possibilities. Yes.
So, I know how you had talked about where you looked at delta and didn't see it being significantly more hydrophobic
Right.
amino acids that are in that interface. I don't know. I mean, I guess if you really wanted to know the binding strength, you'd have to do a full QN and free energy calculation on the two proteins. But did you consider at all possibly, do those mutations affect the electrostatic attraction between the two pieces or not?
Yeah, they would. And just looking at what the mutations are, they are becoming a little bit more electrostatic. I think both of them lead to a positive, if I'm not remembering correctly, they both lead to a positive amino acid from one that's not. And so that would create more electrostatic attraction. So potentially hydrophobicity isn't the driving force for Delta in terms of why it was more impactful.
Yeah, but these receptors negatively charged, so
Ah.
it's a positive charge.
Our Zoom attendees are very happy. No, they had no questions. Thank you for joining us. So if you can hear that, and that's fantastic, Bill.
Thank you very much.